Amy joins us from CDER at the FDA, where she's a team lead in the Pharm Tox division, and is going to speak to us about the ICH S5. Thank you.
Thanks, everybody. I want to thank everyone, the organizers, for inviting me to participate, and also just acknowledge that a lot of my slides, bulk of my slides, were from colleagues, Ron Wang, Paul Brown, and I want to thank Nikisa for also helping put this together. And also I want to say that I forgot my disclaimer slide when I was doing this. So this is just so you all know, you can imagine the FDA disclaimer that these are just my ideas and does not represent the FDA or CDER. So I'm going to talk a little bit about how NAMs have made their way, or have been in ICH guidances for quite some time, and then transition a little bit to the ICH S5 guidance on reproductive toxicity. But just so everyone's aware, FDA, everything that we do is based on federal regulations. So, that's kind of the premise of what we do. And in INDs, investigational new drugs, within this Code of Federal Regulations, it clearly states that, we need to have adequate data, information on the pharmacology and the toxicology of the drug to make sure that it's safe to go into human clinical investigations. But, that data can come from animals or in vitro. And that or in vitro has been in the regulations for many, 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 many years. It's not something new. And then we have guidances that help sort of explain the laws. But, those are, guidances, recommendations. And also, just so everyone is aware, the FDA, we do not require animal studies. They are warranted in certain situations. And a lot of this comes to how we will use data from NAMs, and that maybe when data from NAMs, could supplement or potentially replace animal studies, or when maybe animal studies would still be warranted because we wouldn't be able to get the necessary information otherwise. And then this touches a lot on the topics today of context of use, and that, we at the FDA will be willing to accept data from NAMs when we feel like the alternative approaches are adequate, depending on the context of use, and we'll hear more about this, what is the questions that is being addressed, what data are available to demonstrate that the method is valid for addressing that particular context of use. And then, we're going to touch a little bit, about these, how alternative assays are described in various guidances. Before I get into S5, the reproductive guidance, I want to take a step back and talk first about the ICH M3R2 guidance, which hopefully many are aware. This is sort of our Bible for non-clinical studies. It talks about everything and anything, but right on the very front page of this guidance, it clearly states that, you can use alternative approaches, right there in the guidance, and encourages people to discuss these approaches with the FDA. And again, in the objectives, it clearly talks about the 3 Rs, and we're open to that, and that consideration should be given to the use of new in vitro models, evaluations for safety evaluation. Now a little history. So NAMs, it's not new. We've been using and accepting data from NAMs for quite some time. So I'm going to run quickly through a little history lesson here. So for example, phototoxicity, decades ago, all of these studies were done in vivo, in rats or rabbits or guinea pigs. But, since that time, in vitro assays have actually successfully replaced in vivos. So these are some of our gold standard success stories, you could say, about where NAMs have successfully replaced. So, and then in that, guidances have come about. So the ICH S10 guidance, the phototoxicity guidance, clearly talks about in chemical assays and in vitro 3T3 NRU assays, instead of those animal studies that were done maybe decades ago. Mutagenic impurities is another one, in the mid 2000s when QSAR came about. So now we're using QSAR computational analysis to look at the risk of potentially mutagenic impurities instead of doing maybe other in vitro or maybe in vivo animal models to assess that risk. And again, a guidance has come about that in 2014. Ocular irritation is another great example, where we used to do rabbit Draize tests in vivo and animals. Those are not done anymore. They've been successfully replaced with various in vitro assays. And here we mentioned the OECD test guideline. And I think this is sort of the area where, it helped that these OECD testing guidelines are available. And that kind of helps us as regulators feel confident in that the assays are doing what they're supposed to doing. And we have confidence in the data if there is an OECD guidance out there for how to conduct that particular study using that NAM. Not to say that that's a requirement, but that definitely helps with our level of confidence. Dermal irritation, this is sort of, I think, my last example. Again, same Draize skin tests were done in animals years ago. And now we have various in vitro assays, including a more recent one of a 3D reconstructed human epidermis models are acceptable now, although we haven't seen, we have actually seen an increase in those. So now we're going to talk about the ICH S5, the R3 revision guidance from 2020. And this is the detection of toxicity to reproduction for human pharmaceuticals. And what's interesting is within the R3 guidance revision, it describes alternative approaches that could be used to assess embryo-fetal development risk for a drug, biologic, or a vaccine. So some of the guidance provides actually some good information and performance criteria if someone were to use a alternative approach to assess embryo-fetal toxicity. And then scenarios where these assays could be appropriate and where you could use those, and then how they fit into the overall risk assessment for a new compound. So what are the approaches that are outlined in this guidance? It clearly says that, if they are properly qualified, these alternative assays, have the potential to either defer or replace, in certain circumstances, these conventional in vivo rat and rabbit embryo-fetal development toxicity studies. However, the big however is these assays need to have that level of confidence, for the human safety assurance that, these assays are at least equipped. We always say this middle sentence is, I think, something that you hear a lot, is your NAM assay needs to be either equal to or better than the existing if you want to replace something. So I think that's the big, how do we get there? How do we get that level of confidence that this assay is equivalent to the in vivo assay? And then also, it's not going to be a one-for-one replacement. The idea that, for regulatory purposes, you may have multiple NAMs put together, as a series of tests to maybe replace a standard in vivo assay. And then since we're talking globally here, I just want to point out that this is an ICH international guidances, but there are some clear differences between what is recommended for the FDA versus outside the US. So for example, embryo-fetal development studies can be deferred, but outside the US in specific circumstances. So it says that a qualified, again, alternative assay could be that predicts the outcome in one species. It can be combined with a preliminary embryo-fetal development assay in a second species to allow up to 150 women of childbearing potential for up to 3 months in a clinical study. This is outside the US. And why do we say outside the US? Because in the United States, for the FDA, we actually can defer all of the embryo-fetal development testing until phase 3 for the most part, unless there's a specific cause for concern. So there are some distinct differences in the guidance that way. So if you're not familiar with this guidance, I highly recommend you take a look at it, especially Annex 2. So Annex 2 is a pretty detailed kind of step-by-step guideline for how, kind of a qualification framework for developing an alternative assay to look at embryo-fetal risk. It does not specifically point out or recommend specific assays. It's more of a general guideline. And then again, under certain circumstances, if these assays are qualified, they can be used to support hazard ID and risk assessment. In certain circumstances would be where the adverse effect is likely. So the mechanism of action of the drug is such that it's a teratogen, by the class that that class of drug is a teratogen. So that might be a good situation where we could maybe use a alternative assay to say, yes, in fact, it is a teratogen. So we don't need to do an animal study. If there are equivocal findings in an animal study, maybe an alternative can be used to help figure out which way it's going to go for equivocal findings. I already talked about to support the inclusion of women of childbearing potential in ex-US clinical studies. And of course, the last one, which I think is more universal in my opinion for the use of a lot of these NAMs, is the consideration of using an alternative assay for pharmaceuticals that are being developed for severely debilitating and life-threatening diseases. Also in the annex, it does not specifically state any particular assay or recommendations for any particular assay, but just some basic scientific principles. And also just noting that if one regulatory agency accepts the assay does not necessarily mean that another one will. So that's a big, just keep that in mind. And that the guidance is going to be subject to kind of being maintained over the years. So they're not going to have to reopen the guidance and come up with a whole big new revision, every time they have more information to add to it. But as the science evolves, regulatory acceptance evolves, we can sort of update and get more information into this guidance on that. So what are some of the specifics in the annex? So there are, okay, thank you. I think I have 2 slides left. Okay. So some of the specifics, talking about a qualification to identify malformations and embryo-fetal lethality, MEFL is what we talk about. It talks about, the model, biological plausibility, how the, details of how the algorithm was employed for looking at the positive and negative controls. How is the algorithm described for relating the concentrations in vitro using your assay to the in vivo exposure? That's a big one. How do you make that translation? And do you have adequate data and information in there to justify that if you're going to be making safety margin recommendations? And then lastly, it contains a list of compounds that, you need to use in your assay to identify, the positive compounds, the negative compounds, and identify NOELs for the methyl. And then of course, other compounds can be added with justification to support qualification of that. I ran through that really fast, but I'll be happy to take questions at the panel discussion.